comes science. They might be trying to here comes science.
Hi, Dr. H here. This video is going to be a short review of the scientific design and analysis aspect of the AP Biology test. So we'll start right off with some parts of an experiment that you should all be familiar with. First off, we have our hypothesis. A hypothesis is our predicted or proposed explanation of an observation. And the two most important aspects of any scientific hypothesis are that it must be testable and it must be falsifiable. Okay, we have to be able to objectively collect data to investigate our hypothesis and to test whether or not it is valid. And we must be able to prove our hypothesis incorrect. Looking at some different types of hypotheses that we may encounter, first we have our null hypothesis. And this is sometimes a little tricky to wrap our heads around, but this basically is a statement saying that our two variables, our independent and our dependent variable, which we'll talk about in a minute, do not have any relationship between them. So there is basically no effect of changing one variable on the other variable. Okay, this is usually where we start. Okay, we start with this hypothesis, and if we disprove this null hypothesis and we determine that there is some relationship between our two variables, then we come up with some sort of alternative hypothesis. And this alternative hypothesis would then indicate what type of relationship we see between our two variables. So what are these two variables that we will have in our experiment? They are the dependent variable and the independent variable. The dependent variable is whatever we are measuring within our experiment. When you read a question on the AP test, they generally will very clearly state what was being measured. The independent variable is whatever the experimenter is changing or the experimenter is controlling within the confines of the experiment. Okay, another very important aspect of experimental design is the controls. Okay, what types of controls does this experiment contain? You may also be asked to evaluate the controls. Were these the correct controls? Or maybe to propose additional controls. Okay, usually with this type of question, they are referring to what we call experimental controls or the control group. Okay, and in this case, the control group is a set of experimental subjects that are exactly matched to our experimental group except for our one independent variable. So this control group is very carefully set up so that everything else, every other aspect is exactly the same so that the independent variable can be isolated and adequately determined if this is affecting the dependent variable. The other way that we use the term controls is with our positive and negative controls. And these are included to make sure that whatever test we are doing, whatever procedure we are using, is working correctly. So a positive control is a test that we know will give us a result. A negative control is a test that we know will give us no result. So this is more of an internal check on our procedure and all of our equipment and reagents to make sure that they are all working correctly. So moving on from experimental design, how do we report out the results of our experiment? Very often on the AP test, we will see graphs, and we will need to maybe create a graph or analyze a graph. This year, for the 2020 AP test, you will not be asked to create a graph, but you may need to do some analysis of a graph. So what type of graph is correct for our data? Basically, we will see line graphs or bar graphs. Okay, a line graph is most useful when we have a constantly changing independent variable on the x-axis. So a line graph then will allow us to interpolate data points. In between points on our x-axis, we can find the relationship. Line graphs are also used to help to visualize very small changes. A bar graph, on the other hand, is more useful when we have independent categories for our independent variable, where we don't have a constantly changing variable. It's different treatment groups or vastly different time periods. Bar graph can also be used when we have very large changes in our variables, and this can make it a little bit easier to visualize. When we plot these independent and dependent variables, the independent variable will go on the x-axis, and the dependent variable will always go on the y-axis. So sticking with graphs a little bit and looking at some analysis that you may be asked to do with graphing, you may be asked to evaluate some trends that we see between our two variables. So what type of relationship exists between the dependent and the independent variable? A direct relationship means that as one variable goes up, the other variable goes up. Or as one goes down, the other goes down. A direct relationship would be both variables changing in the same direction. An indirect relationship is the variables changing in opposite directions. So as the independent variable goes up, the dependent variable would go down. Some calculations that we may need to make. The main one I think that we would see on the AP test would be calculating the rate of change. Okay, so this would be for a graph that has time as the independent variable on the x-axis. Rate calculation, remember, that would just be the slope of the line, which would be found by the change in y divided by the change in x, or y2 minus y1 divided by x2 minus x1. Just pick 2 points on the graph, find their values, get as close as you can, depending on how the graph is presented, and find the rate. Oftentimes in the question, they will give you the 2 time points to find the rate between. If they don't give you the time points, then just pick 2 that will work easiest for you. For the most part, there will be some variability in the accepted answer, allowing for difficulty in actually reading the exact numbers off of the graph. Okay, and then the final aspect of graph analysis are the presence of error bars. Okay, error bars are plotted using either the standard error or the standard deviation. These 2 measures give us slightly different information about our data set. The standard error is used to compare our experimental group to the larger population as a whole, and the standard deviation is used to measure variance within our small group. So both of these error calculations are very useful and can give us a little bit more information about our experiment. If a graph has error bars, it should indicate which is being used. For our purposes, it's probably not going to matter too much which one is actually being plotted. The important point is, what does that mean in terms of comparing two data points. The main thing we need to look for there is, do our error bars overlap? If the error bars are overlapping, that is a good indication that those data points are not significantly different. We want the error bars to show a little bit of space between them before we can really say that these two means, these two averages, are actually statistically different from each other. So that is it in terms of scientific design and analysis. So hopefully that cleared some things up, and I hope to see you guys again soon.
Science like Galileo dropped the orange.